The Environmental Molecular Sciences Laboratory, or EMSL, is a Department of Energy, Office of Science user facility sponsored by the Biological and Environmental Research Program. Scientists from around the world can access EMSL's world-class expertise and resources for free through a competitive peer review proposal process. EMSL staff and users seek solutions for the most critical energy and environmental challenges facing our nation and the world. Clean and reliable energy, improved carbon storage capabilities, and controlled biological transformation of environmental contaminants. Through EMSL's integrated research platforms, users can harness the power of EMSL's multidisciplinary expertise.
Hi, I'm Scott Lee, the lead for the Structural Biology IRP. Our goal is to obtain structural and dynamic information on proteins and other biomolecules at nanoscale, spatial, and temporal resolutions to infer function. Behind me is our Aquilose CryoFib SEM, which we use for 3D tomographic imaging and for sample prep for cryo-TEM. This lab houses our Krios cryo-TEM, which we can use to obtain atomic scale structure of proteins and protein complexes. Along with our NMR, mass spectrometry, and atom probe tomography capabilities, these instruments provide a holistic understanding on the structure and dynamics of biomolecules and yield insights into their function.
Hi, I am Mary Lipton, the Biomolecular Pathways IRP lead. We use multi-omic approaches to map responses of biomolecular pathways to environmental changes. Seen here is the NMR instrumentation used for quantitative metabolomic experiments. We use autosamplers on every instrument to remove the need for manual sample changes and increase throughput. In our advanced separations and mass spectrometry laboratory, lipids, metabolites, or proteins are analyzed in multi-omic experiments. Each instrument is completely automated and run 24 hours a day, 7 days a week, producing over 1 terabyte of data per month. These data are then integrated to produce metabolic and regulatory maps of our biological systems.
Hello, my name is Jennifer Hurley, and I'm an Associate Professor of Biological Sciences at Rensselaer Polytechnic Institute. I've worked with EMSL since 2014 to utilize their mass spec, sequencing, and NMR facilities to characterize circadian output in order to demonstrate that post-transcriptional regulation is essential to metabolic control from the circadian clock. This collaboration has led to several high-profile papers, including one in PNAS and another one in Cell Systems, and has also led to the joint funding between the NIH and the DOE of a U01 grant to model how metabolic output is timed by the circadian clock.
Hi, I'm Alex Baliev, the lead of the Cell Signaling and Communications IRP. Our goal is to characterize signaling pathways and capture molecular trafficking that underpin the dynamic behavior of multi-organism systems. To accomplish this, we use fluorescence-activated cell sorting and laser capture microdissection to isolate individual cells or unique subpopulations based on their size or optical properties for downstream phenotyping analyses. Enabled by next-generation single-cell sequencing and nanopods proteomics, we capture micron-scale heterogeneities in gene and protein expression profiles, which otherwise would be lost using traditional bulk analysis. Single-cell molecule and super-resolution fluorescence microscopy that helps us visualize these processes in live cells with nanometer-scale resolution, shedding light on the dynamics and mechanisms of interactions within complex microbial systems.
Hi, I'm Emily Graham, the lead for the Biogeochemical Transformations IRP. Our goal is to understand the interplay of geology, chemistry, and biology in the environment. Behind me is our new 7T FTICR-MS, which is a key instrument in the soils automation pipeline. We also have technologies that analyze spatial characteristics and mineralogies at sample. One example is the Quantum 2000 X-ray photoelectron spectrometer, which generates information on surface mineralogy at the micrometer scale. Along with tomography and microscopy, these instruments provide a holistic understanding of biogeochemical systems.
Hello, my name is Rene Boiteau, and I'm an assistant professor at Oregon State University. My research focuses on the ways in which plants and microbes dissolve and take up metals in soils. So emsl is pioneering much of the technology and the informatic development that enable the major advances in our research, and I view our partnerships with emsl as essential to our science. The metabolomic and metabolomic and proteomic capabilities of emsl enable us to identify the specific metabolites and genetic pathways that are involved in metal cycling. And then the unique imaging capabilities like TEM, helium ion, and confocal microscopy allow us to understand how microbes and plants physically interact. And we also work with emsl staff using molecular imaging such as nanosims and multi-mass spectrometry to localize specific metals and metabolites across microzones and soils. And then finally, we use spectroscopy expertise, including NMR and Mossbauer, through emsl to structurally characterize novel metabolites and to understand the composition of minerals and organic matter in the environments that we're studying. So combining these advanced analytical approaches with emsl expertise has become central to the research in our group. These partnerships help us to develop predictive frameworks of how micronutrient and contaminant metals are transported in the environment, and ultimately how they affect water quality, carbon cycling, crop growth, and climate.
Ecosystem Interface's instrument research platform truly spans ecosystem scales to help interpret relevant fluxes, transport rates, and processes. In support of this, we maintain cross-cutting instrumentation to enable innovative science. For example, the Subsurface Flow and Transport Lab provides a physical model for detailed analyses and investigations. To help complement this, the isotope ratio mass spectrometry capability enables quantification of relevant transport and reaction rates. And at a much finer scale, NanoSims enables highly resolved spatial analyses at the scale of geochemical and microbial processes. Taken together, we can use these and other tools to better understand fluxes and processes linking different ecosystems.
Hi, I'm Amir Ahkami, the lead for the Plant and Ecosystem Phenotyping IRP. We aim to dissect the plant genotype-environment interactions to understand the molecular mechanisms that control feedstock productivity. To accomplish this, we use the plant ecosystem lab facilities, including plant growth chambers and image-based phenotyping capabilities like dynamic environmental photosynthesis imager or non-invasive automated root imager. We also use a stable isotope probing approach to investigate carbon and nutrient flow at the plant microbe interface. Along with obtained information at the molecular and cellular levels, these facilities provide a better understanding of plant and ecosystem traits at the system scale for advancing bioenergy crop and ecosystem sustainability.
I'm Jay Bardhan, lead scientist for the Systems Modeling and Data Sciences IRP. This is EMSL's newest supercomputer, Tahoma. Tahoma is a heterogeneous machine, meaning that it has two kinds of nodes. In addition to 140 traditional CPU-based nodes, it has an additional 24 nodes, each with two powerful GPUs and lots more memory and disk space. The team designed it to meet diverse BER mission needs from the atomic scale to the watershed scale. What really makes EMSL computing unique as a resource are the subject matter experts and high-performance computing teams who work together to help users translate science needs into high-performance solutions.